All right, now it's time to connect what we learned previously about the cell cycle and mitosis with what we're learning about now, heredity. And the connection has to do with meiosis, not mitosis, but meiosis, which is a special kind of nuclear and cell division that produces gametes. And what it does is it shuffles and recombines genes from two parents to produce a new individual that is different from both those parents. So as Mendel discovered, during the production of gametes, gametogenesis, genes are segregated and assorted, another way of saying shuffled, into gametes, and at fertilization they are then recombined into offspring in such a wide variety of possible combinations that no two offspring are alike. And that's all because of the process of meiosis. But before we get into gametogenesis by meiosis, we need to learn a little bit more about chromosomes. We've already learned some about chromosomes, but we need to learn a little bit more about chromosomes. So the first thing is that different species have a different number of chromosomes. So for example, humans have 46. Other apes, and I say other apes because humans are considered within the family of apes, other apes have 48. So like chimpanzees and gorillas have 48. Fruit flies, as you can see here, have 8. Right? So we're looking at 4 pairs of 8 chromosomes in fruit flies. And the thing to note about that is that the number of chromosomes that a species has no connection to how complex it is. All right, so there's no relationship between the number of chromosomes and the complexity of the organisms, because some plants have hundreds of chromosomes, and you may not consider plants to be more complex than humans, which only have 46. So as I mentioned, what we're looking at here is Drosophila, which is the genus of fruit flies, and they have 8 chromosomes, 4 from each parent, just like you get 23 from each parent to total 46. And your 46 chromosomes, just like these 8 chromosomes for Drosophila, fruit flies, are in pairs, and they're known as homologous pairs, homologous chromosomes. So there's one homolog, one chromosome from each parent, and they pair up, they match up. They have the same genes on them. So they're the same in length, they're the same in banding pattern, because when you stain them, they get light and dark bands on them, and you see the same pattern of bands on homologous chromosomes. They have the same centromere location, they're the same length, same size. Yeah, I already said that. But they can have different alleles. So even though they have the same genes, they can have different alleles. They may not, they may have the same alleles, but they can have different alleles. That's how you get homozygous dominants and homozygous recessives and heterozygotes, depending on which alleles are on those 2 chromosomes. So there are 2 general kind of cells in your body, and some of those cells are diploid and some of those cells are haploid. And we've already come across this term previously, so hopefully you're already very familiar with it. Diploid means the full number of chromosomes. That means 2 sets. So again, just like you got 23, a set of 23 from your mother, a set of 23 from your father, and that totals 46. That's your diploid number. The diploid number of chromosomes in human beings is 46. So 2 complete sets in humans of 23. In fruit flies, it's 2 complete sets of 4 to total 8. 1 from each parent. So N in 2N, in the symbols here for diploid, that N represents 1 set of chromosomes. So there are 2 sets, right, in diploid. In animals like Drosophila and other organisms, the somatic cells are the ones that have the full number of chromosomes that are diploid. So somatic refers to body. Soma refers to body. So these are body cells. And all your body cells are diploid. They have 46 chromosomes. And again, the diploid number for Drosophila is 8. So all the body cells of this fly, and that's basically everything that you can see, the cells that make up everything that you can see all have 8 chromosomes. Haploid is 1 set of chromosomes. So in other words, for humans, that would be 23, 1 set. So In sexually reproducing organisms, only gametes are haploid. So in your body, if you're a male, sperm cells are haploid. They have 23 chromosomes. If you're female, then eggs are haploid, 23 chromosomes. Because when a sperm fertilizes an egg, they need to combine into the full diploid number. And again, in humans, that would be 46, right? So 23 plus 23, 46. And that's what you have in the one cell that results from fertilization. When a sperm fertilizes an egg, the zygote that results, the one cell that results, that's known as a zygote, and the zygote has 46 chromosomes. And that's how you and I and everybody started out, as a zygote. Gametogenesis is by meiosis. The formation of gametes is by meiosis. In other words, the way that sperm and egg get only 23 chromosomes is by the process of meiosis, special form of nuclear and cell division. So how do we visualize chromosomes or see chromosomes, and how can we tell how many chromosomes are in a cell, any particular cell? Well, we can do what's known as a karyotype for people, and we do for people, and we can do it for other animals too. But a karyotype is an ordered arrangement of chromosomes, just like you see here. An ordered arrangement of chromosomes from one cell, and they're arranged in homologous pairs. So each pair here is a homologous pair. And these are my daughter's chromosomes. Kayla Zagre, that's her name. And we got this karyotype done before my daughter was born. So we were able to see her chromosomes even before she was born. And the reason we did this was to check to see whether she had Down syndrome. Because when my wife was pregnant with my daughter, she was 35 years old, which is on the older side. And the incidence of Down syndrome, in other words, the probability of Down syndrome, increases with the age of the mother. So the older the mother is, the higher the possibility of Down syndrome. So we had this karyotype done for our daughter before she was born to see whether or not she may have Down syndrome. So again, it's an ordered arrangement of chromosomes in homologous pairs according to size and banding pattern. So you'll notice that one homologous pair, the two chromosomes that make up that homologous pair, they're the same size. And it's hard to tell here, but they also have the same banding pattern, light and dark bands on them from being stained. So of course it goes without saying, because this is my daughter's, this is a human karyotype, and it's a normal female karyotype. Normal meaning that she does not show that she has did not show that she has Down syndrome. Female because the sex chromosomes are right here. So all the other chromosomes are known as autosomes, and the autosomes are all the chromosomes other than the sex chromosomes. So again, this is pointing out simply that this is one homologous pairs of 23. There are 23 homologous pairs of chromosomes here. So the diploid number for this species, in other words humans, is 46, 23 pairs. Haploid is 23, no pairs. So if we were to do a karyotype for a sperm cell or a karyotype from an egg cell, we would see that there would be only one chromosome at each of these positions. At all of these 23 positions, there would only be one chromosome. And so the sex chromosomes here are the 23rd pair. The number's not on here, so I added it. So the way that the way this was done is through a process known as amniocentesis, and I was there for the procedure, and what it involves is putting a big long needle into the womb of the mother, through the abdomen and into the womb, and through the amniotic sac that surrounds the baby and into the fluid that the baby is floating in, and there are cells from the baby floating around in that fluid. And so they draw out some of that fluid, and they look through the fluid and they find a cell that is mitotic. In other words, it's undergoing mitosis. And you know that that's a cell that's going to have condensed chromosomes. The chromatin would have been condensed into chromosomes. So these are so they cut the cell open and spill out the chromosomes, and that's what we're looking at here. This is a picture of the cell that they found and spilled the chromosomes out of. And then in the old days, they used to actually use scissors. They would print out the picture and then use scissors to cut out the chromosomes and then place them into order according to size and banding pattern in homologous pairs. Today, it's all done digitally, but the chromosomes that are shown in this karyotype are the same chromosomes that are shown in the picture down here. So a mitotic cell was obtained, broken open to spill out the mitotic chromosomes, and then they were arranged into order by size and banding pattern. And that's a karyotype. So just to add some labels here, that's a mitotic cell, and these are mitotic duplicated chromosomes. And these are all mitotic duplicated chromosomes from the cell below, from this cell. Like I pointed out, chromosomes 1 through 22 are all known as autosomes. The sex chromosomes are known as sex chromosomes. So all chromosomes that are not sex chromosomes are known as autosomes. Now you can't really tell by looking at these, but they are duplicated chromosomes, so that means that each one is made up of 2 sister chromatids, right? If you'll recall. And they're attached at the centromere. Now they're like attached along their length, so they're not really separate, so you can't really see the 2 separate chromatids that are here, but you can kind of tell in the banding pattern that there are 2 chromatids side by side, kind of connected all along their length. And for each homologous pair, for each one of these pairs of chromosomes, one chromosome came from my daughter's mother, in other words my wife, and one of them came from me. There's no way to tell by looking at them, but that's how it is. So my daughter is my daughter and not my son because she has 2 X chromosomes. That's how the sex chromosomes determine gender. 2 X chromosomes is female, and if there was 1 X and 1 Y chromosome here, then my daughter would not be my daughter, she would be my son. And the last thing I want to note here about the Y chromosome is that it's much smaller than the X chromosome. Much smaller, and it doesn't have all the genes that are on the X chromosome. But it's the Y chromosome that makes a male, that turns a male into a male. So this is a normal female karyotype, 46XX. And it's because of that, it's a girl. And this is the first that we knew it was a girl. So now we'll get into the process of meiosis. And if this diagram looks familiar, it's because it's very similar to what we were looking at back when we were studying mitosis. But meiosis, and especially meiosis I, is different than mitosis. So meiosis is nuclear cell and cell division that reduces one diploid somatic cell, that's what we have right here, one diploid somatic cell, to 4 haploid gametes by separating the homologous pairs. Now, we're not seeing 4 haploid gametes here because we're only looking at meiosis I. This is the first round of division, and there's another round of division, this meiosis II, that's going to follow this. And it's the separation of homologous pairs that happens during meiosis I here that reduces the number of chromosomes in half. So if you'll notice here, we're starting out with a cell that has 4 chromosomes in the diagram, and by the time we get to the end of meiosis I, we have 2 daughter cells that only have 2 chromosomes in them. So the reduction in chromosome number happens during meiosis I, which is then followed by meiosis II. So there are 2 rounds of nuclear and cell division involved in meiosis. And the first is meiosis I. So duplicated chromatin condenses into duplicated chromosomes. That's what's happening here as we go from interphase 1 to prophase 1. And by the way, you have to include the 1. When you're talking about meiosis I, you have to include the 1. And when you're talking about meiosis II, you have to include 2 for the different phases, the names of the different phases. So the chromosomes from homologous pairs line up along the metaphase plate. Homologous pairs, they get together in homologous pairs, which right there is different than mitosis. And then they line up along the metaphase plate together in pairs, which is very different from mitosis. And then the homologous chromosomes separate. During anaphase, the homologous chromosomes are separating. But note that they are still duplicated chromosomes. They're still made up of sister chromatids. And like I mentioned already, they end up in two haploid daughter cells by the time we're at the end of meiosis I. So all of that is not mitosis-like. When it comes to these 2 parts of meiosis, meiosis I and meiosis II, meiosis I is the one that is not mitosis-like. Meiosis II is very similar to mitosis, so it is mitosis-like. So all the differences between mitosis and meiosis happen during meiosis I. So let's take a look at what's going on here in greater detail. So here we have diagrams representing interphase and prophase I. So this is a somatic parent cell. This is the nucleus with duplicated chromatin, right? And we also have duplicated centrosomes, which have centrioles in them, because we're looking at an animal cell as our example here. So centrosomes with centrioles. So interphase I is the same as interphase preceding mitosis. In other words, we go through G1 and S and G2, and by the time we're at the end of G2, everything is doubled. The DNA has doubled. If you recall, it gets doubled during the S phase, because S stands for synthesis of DNA, and we get double everything else. All the enzymes are doubled, all the organelles are doubled, so we're ready to divide. And then we get into prophase I, and we see the spindle being formed between the centrosomes that are moving towards opposite poles of the cell, just like in mitosis. But what's not like mitosis is that homologous pairs pair up, and they're duplicated chromosomes, so that's similar to mitosis. But pairing up, that's totally different than mitosis. In mitosis, the chromosomes do not get into homologous pairs. So somehow these homologous pairs get together, and they form these structures that we call tetrads. So tetrad refers to a homologous pair of chromosomes that have come together, that have paired up. And you'll notice that the chromatids from these two duplicated chromosomes are crossing each other. They actually come in contact with each other and cross each other, and that's when crossing over, an event that we call crossing over, occurs. And we'll be looking at that in greater detail on the next slide. So in prophase I, duplicated chromatin condenses into duplicated chromosomes, and the nuclear envelope breaks down, and the centrosomes move towards opposite poles, forming the spindle. So that describes what's going on here in prophase I. Homologous chromosomes pair up, and them getting together is called synapsis. So the term synapsis refers to the coming together of homologous chromosomes. And that's literally what synapsis translates into. It means coming together, or come together. So that forms tetrads, 4 chromatids. The reason they refer to these as tetrads is because there are 4 chromatids. Tetra means 4. So these are tetrads because of those 4 chromatids. And they exchange genes through crossing over. That's what happens when the chromatids from these 2 homologous chromosomes come in contact with each other. Pieces of them break off and exchange between the chromatids. That's called crossing over. And again, we'll look at it in greater detail on the next slide. The thing that I want to note about crossing over before we look at the details is the crossing over is the rule, not the exception. It kind of seems like it's a mistake or an accident because these chromatids are breaking off and then reattaching to other chromatids, but it's actually more frequent than not. It's the rule, not the exception. So crossing over can also refer to it as recombination because we're recombining DNA from 2 different chromatids. This occurs during prophase I. And it definitely does not happen in mitosis. So we only see this it only happens between homologous chromosomes, and because homologous chromosomes don't pair up in mitosis, crossing over doesn't happen in mitosis. So when homologous chromosomes pair up, in other words, synapses come together, forming tetrads, the non-sister chromatids, and that's what we're looking at here, chromatids that are not on the same chromosome are referred to as non-sister chromatids. If you'll recall, they're sister chromatids when they're making up the same chromosome. But if they're on different chromosomes, they're referred to as non-sister chromatids. They'll cross over, forming chiasmata. Chiasmata means cross, or chiasma means cross. So chiasma is one cross. Chiasmata refers to multiple crosses, so it's plural. Chiasmata is actually plural of chiasma. So this is a chiasma, a cross of 2 chromatids, 2 non-sister chromatids, and they're exchanging homologous segments. In other words, you'll notice that they're exchanging the same letters here. And these letters don't represent genes. They represent whole sections of chromosome. right? So they're exchanging the same sequences of DNA. Remember that these chromatids are made up of DNA, so they're exchanging the same nucleotide sequences of DNA. The same genes are crossing over here through the process of crossing over, and they're recombining. So we call this recombination, genetic recombination. So let's go ahead and add some labels to the diagram here. So we've got a homologous pair of chromosomes. They're together in a tetrad, so they've undergone synapsis, they've gotten together, and that's when crossing over occurs. When they get close enough to each other, their chromatids actually physically cross and touch each other, and non-sister chromatids will form a chiasma, a cross, and exchange pieces of each other. And they're exchanging the same pieces of each other with the same genes. They may have different alleles, but they're the same genes. So like I already pointed out, the letters on the chromatids represent chromatid segments, whole segments that might have hundreds of genes. So many genes are crossing over here, not just 3. So it's not, again, the letters are not genes. So many more than just 3 genes are crossing over here. And the DNA is being recombined. So it's recombinant DNA. And again, as I already pointed out, crossing over or recombination is the rule, not the exception. This happens a lot between homologous chromosomes. And the significance of that is it really shuffles the genes, right? The more you shuffle the genes, the more variation there is in the gametes that are being produced through the process here. And the more variation there is in the gametes, the more variation there's going to be in the offspring that result from those gametes. Now, let's relate this to the principle that Mendel discovered of independent assortment, because you would think that these genes being on the same chromosome would always be inherited together. But they're not because of crossing over. And because crossing over happens so frequently, it's more often than not that genes get separated. Even though they're on the same chromosome, they get separated and as a result of crossing over. So even though they really are together on the same chromosome, and therefore you would think they were linked, they actually get separated by crossing over and appear to assort independently. And again, as I already pointed out, because I can't help myself, the ultimate result of crossing over is an increase in variation, an increase in shuffling of the genes, which increases variation in the gametes. And also at the population level, there's a lot of variation between individuals, and you know the significance of that. It's the raw material of natural selection and evolution. Variation is where evolution begins. It's where what natural selection acts on and allows species to evolve. All right, so that all happens during Meiosis I, or actually prophase of Meiosis I, so prophase I. Now let's take a look at the rest of So here's metaphase I, and it looks very similar to what we see in mitosis, except for the fact that the tetrads are still formed. In metaphase, the homologous chromosomes are still paired up, and they're lined up along the metaphase plate, paired up. So homologous pairs of duplicated chromosomes, they're still in tetrads, in other words. Crossing over has occurred, so you'll notice by the color coding here, there's a little piece of red on the yellow and a little piece of yellow on the red, same with the green and the blue. So crossing over has occurred. In anaphase, the duplicated chromosomes, so they're still made up of 2 sister chromatids, separate. Remember, ana, it means away, and so the chromosomes move away from each other. But what's separating here are not the chromatids. In mitosis, it's the chromatids that separate and then become unduplicated chromosomes. Well, here, it's the homologous chromosomes that are separating. And so they remain duplicated chromosomes after they separate. So during metaphase I, homologous chromosome pairs in tetrads line up together, one of each pair on opposite sides of the metaphase plate. So that's what we're seeing here. And the chromosomes line up randomly. And that's significant. I've got that in all caps and bold because that's significant, because that also results in an increase in variation through this process of meiosis that increases the variation in the gametes, because it's totally random how these chromosomes line up during metaphase. So it results in greater variation. And then in anaphase 1, homologous duplicated chromosomes separate. So they're still duplicated chromosomes. That's why duplicated is all in caps. And that represents the reduction in chromosome number. So if you're asked, for example, when during the process of meiosis the reduction in chromosome number occurs, you'll want to answer anaphase 1, because that's when the homologous chromosomes separate. So these new daughter cells that are about to be formed are going to have half the number of chromosomes because of the split, because of the division, because of the separation of the homologous chromosomes during anaphase 1 of meiosis I. So the resulting daughter cells at the end of meiosis I will be haploid. They'll have half the number of chromosomes, and in this diagram that means 2 chromosomes instead of the 4 that the cell started out with. And that brings us to telophase I and cytokinesis I. So again, just to remind you, these are still duplicated chromosomes. So unlike mitosis, when we're at telophase of mitosis, they are unduplicated chromosomes. But this is meiosis, the end of meiosis I. So we still have duplicated chromosomes made up of 2 sister chromatids. So in telophase I and cytokinesis I, they occur pretty much the same as mitosis, except that the resulting daughter cells are haploid, only have 2 chromosomes in this diagram, and they're genetically different because of crossing over. But they still contain duplicated chromosomes, so there's still something to divide up, and it's the same thing that gets divided up in mitosis. So this is the end of meiosis I. Now we're going to take a look at meiosis II, or next time we'll take a look at meiosis II, and you'll see that meiosis II is very mitosis-like. It's very similar to mitosis. So on to mitosis II.